Welcome back to the course Fundamentals of General Immunology. So today we'll discuss about some of the interesting facts on the topics which we have covered earlier, mostly towards the discoveries that caused the landmark understanding on the field of immunology. So if we look into the prizes given in the science, one of the very prestigious award is Nobel Prize, and this prize were given to the scientists who contributed significantly to the field of science. And the inception of the award was began from 1900 onwards. So the first Nobel Prize in the physiology and medicine is given to a scientist called von Behring, and von Behring got a Nobel Prize on physiology and medicine for his discovery towards understanding the passive immunization. Now, he, along with a scientist, Japanese scientist named Kitasato, he injected animals with a weakened form of diphtheria toxin. Now, diphtheria is a bacterial disease, and the toxin which are produced from that bacteria can cause a diphtheria disease condition. So what Kitasato and von Behring did is they inoculated animals with a weakened form of diphtheria toxin, and once the toxin inoculated into the body produced some milder form of the antibody, and those antibodies, or antitoxin they called at that time, was sufficient to protect the animals from the disease condition if given to some other animal. So essentially what they identified that there could be some kind of material that is produced upon the vaccination or upon the inoculation into an animal that can protect the other animals when they are giving passively the blood to the other animal. So this work led to the foundation of a proper branch of immunology, and particularly what we understood is passive immunization is what he referred to at that time. So there are two basic branches which we discussed in immunology. One is passive immunization, where a ready-made antibodies are given to the patient who are actually suffering from a disease, or otherwise we can try to educate the immune system of the body by providing something from outside so that the body can produce an immune response. And once they are producing an immune response, after getting educated through the external antigen, those are called as active immunization. So there are two branches of immunization altogether, active and passive. So the contribution of Behring goes to passive immunization, where he conducted an experiment to showcase that antitoxins are actually important therapeutic intervention to control the diseases such as diphtheria. So this was the first one awarded to a person who contributed significantly to the field of medicine and physiology. After that, there are many awards given to the field of immunology, and we will try to understand few of them to see that what are the landmark discoveries that happen in the field of immunology. So after 1901, the second one goes to Matnikov and Ehrlich. Now these are work on basically immunity, but Matnikov discovered a very specific cell type called phagocytic cells. Now phagocytosis, we know that a property of a cell that can engulf a bacteria and protect the body from bacterial disease condition. So this is actually the establishment of phagocytosis. So Matnikov discovered this phenomena of phagocytosis along with Paul Ehrlich, who basically gave a concept of humoral immunity, the antibody theory, and because of that they were awarded a Nobel Prize of Physiology and Medicine in 1908. Followed 1908, in 1919, Baudet got a Nobel Prize on discovery related to immunity, and his contribution goes towards the finding very specific proteins which are present in the serum that can help to protect the body from disease condition, and one such protein is complement. So the complement protein, we have discussed that can lead to, say, activation of a cascade of immune response that can neutralize the pathogen from outside. Then in 1960, Burnett and Medawar got a Nobel Prize in acquired immunological tolerance. Now, we basically understand the transplant biology by looking into the tolerance level of the body. Say once the organs are transferred from one individual to other individual, the genetic makeup of the other individual, like donor and recipient we call it, so one who is providing or giving the parts to other people, they are called donor, and the one who is receiving called recipient. So the matching of the donor and recipient biology is what is very important, although that's not a contribution directly to the field, but it was largely done in order to understand the transplant specific immunology. So their contribution goes typically to the transplant immunology. Then in 1980, the Benacerraf, Dowsett and Snell got a Nobel Prize for their outstanding discovery of a cell type that present the antigen to the neighboring immunological cell. We have looked into a protein called major histocompatibility complex protein that can project the peptides or the smaller fragments of the protein to the immunological cells such as B cell and T cell in order to activate the immune response. So their discovery is also very pertinent to understand that how the immune response is activated. Now, if we look into the earlier discovery of Burnett and Medawar about the transplant immunology, now major part of the transplant immunology depend upon the status of MHC of donor and recipient. If there is an HLA typing matched out or the donor with the recipient, then we could see that the success of transplant of an organ such as kidney transplant or the liver transplant is kind of successful. If it is not, then probably we see that there is a failure in most of the cases if HLA type is not matching between the donor and the recipient. So their contribution is outstanding in terms of the tumor, in terms of transplant immunology. Then in 1984, Jörn, Kohler and Melstein got a Nobel Prize. Now Jörn got a Nobel Prize towards understanding the overall immunity, and Kohler and Melstein got a Nobel Prize for discovering monoclonal antibodies. So we will understand the antibody biology and all, but in the last interesting facts we looked into how these monoclonal antibodies are produced and what are the ways these antibodies are used in order to treat some patient who are suffering from some kind of disease condition. So basically they are also utilized for therapeutic intervention in case of infectious disease biology. So after that, in 1987, Tonigawa got a Nobel Prize for deciphering the biology of diversity of antibodies. Now we know that there are many antigens which are encountered by the body every day. There could be thousands, there could be lakhs of different antigen which may encounter in a routine basis in a human body. Each cell type behaves differently towards an antigen. That's a very interesting thing, but people were not knowing how a B cell can produce a separate set of antibody altogether for identifying a specific antigen. So there could be 100 antigen, there could be 100 different kinds of antibody. How this antibody variation happen from a same cell type was not identified. So what Tonigawa did is he looked carefully onto this biology, that how there is a repertoire of an antibody that are generated post immunization or post infection or post antigen entering into the body. And he said that, well, there are many regions in the chain of an antibody genes which are divided into variable domain, diversity domain and the joining domain. So VDJ chains could be present in the gene level, and when there is a transcription, then we see that VDJ chains get arranged into the genetic level in the nucleus, and because of this switching of the VDJ recombination event, we see a variety of different antibodies to be produced by the human body, and that is what the genetic basis of the diversity of the antibody that are generated by the human body. So a great contribution altogether. Still, people are trying to think about how beautifully he documented that individual cell type can behave differently and produce a different subset of antibody because of just a rearrangement of the B and D chains in the antibody gene fragment. After that, in 1996, Dorothy and Zinker-Engel got a Nobel Prize on understanding the cell-mediated immunity. So essentially, we understood the MHC can process the antigen, but then basically the T cell is recognizing the viral antigen only when they are presented with MHC molecule, is what is documented by these two scientists, and they got a Nobel Prize in that. Then after a gap of say 14-15 years, in 2011, the Nobel Prize is given for the discovery of dendritic cells. And basically, one can link the dendritic cell with the innate immunity. So innate immunity is one which we acquired from right from our childhood. So these dendritic cells are the unique internal cells that can process the antigen and display it to the neighboring immune cells. So Butler, Hoffman and Steinman, Ralph Steinman, they got a Nobel Prize for discovery of the dendritic cell and its related biology. And in recent years, 2 more Nobel Prize is awarded to scientists who contributed to the field of immunology. 2018, the award is given to Allison and Honjo for identifying specific checkpoints in the immunological system to treat cancer. So essentially what they identified, that we have the CTLA4 or PD1, which is specifically leading to immunosuppression. And if we can block the CTLA4 and PD1, essentially we can treat cancer patient typically in terms of immunology. So they identify these specific checkpoints related to immune system. And very recently, Bunko, Ramsdell and Sakaguchi got a Nobel Prize for discovery of the T regulatory cell population, which we'll see in our later classes. So the T regulatory cells are specific cell type that can control the immune response by down-regulating the immune system, or we can say that if the body is exaggeratedly responding to an antigen, we can produce a huge amount of antibody, but then there could be something which the body can produce in order to control, and one of the cell type which can be controlling all this phenomena is T regulatory cell. So T regulatory cell population was collectively discovered by Branco, Ramsdell and Sakaguchi. And its related biology. So it's an amazing to see that there are multiple awards given to this field, giving an indication that this is such an important area of research where the field is directly linked to humanity, the benefits of the human health. Unless until we understand immunology, we can't treat a patient or a person suffering from various kind of disorder or the pathological conditions. So we'll try to see some of the unique things about the immunology in this interesting fact. And after understanding this unique way the Nobel Prize is awarded to many scientists, let's try to see very unique things which exist in immunology. And one such concept is called superantigen. We looked into antigen. So these are the proteins which are expressed or produced by various natural ways, and once they are entering inside the body, they can raise an immune response. And those antigens are critical because we can only understand the immune response looking into the antigen. So there is something called superantigen, and as name itself indicates, these are outstanding antigens, and these antigens are actually coming from bacterial toxins. So these are actually a microbial toxin that can cause excessive non-specific activation of the immune system. That means they can, once they are produced in the human body, they can produce a non-specific, so they are not a specific, they can produce a non-specific activation of various immune cells, and therefore they are called as superantigen because they are not under the control of the immune system, so they can just behave differently. By doing this biology, they can do huge bunch of cytokine production in the human body, and therefore we can call those condition as cytokine storm. I think it's a very common term people have looked into at the time of COVID, when we saw during the phases of COVID outbreak at different areas, people died because of massive immune response towards the cytokine production, which we called at that time a cytokine storm. So few of the viruses also can induce a huge amount of cytokine production, and that is what we call cytokine storm. These large amount of cytokines in the human body can lead to toxic shock syndrome. So syndrome is basically the collection of disease condition. They may lead to organ failure such as liver failure, kidney failure, heart failure, or any other organ. They can also lead to systemic blockage of the blood supply, or some other intervention that can lead to systemic shock as well. The examples which we understood till date about the superantigens came from the biology or the research that has been done in some of the bacterial species, such as Staphylococcus enterotoxins. So Staphylococcus is a bacteria which looks like a grape shape structure, and they are known to produce toxic shock syndrome toxin 1, TSST1 they used to call it in short. And there are many other enterotoxin. Now these are known to non-specifically activate the immune system, and therefore they are considered as superantigen. Apart from Staphylococcus, Streptococcus are also important. So streptococcal pyogenes, which can cause specific problems in case of individuals who are suffering from the bacterial disease. Now streptococcal pyrogenic exotoxin, the pyrogenic is the one which can produce a high temperature regulation in the body. So these are some of the other examples of superantigens. Apart from bacteria, some of the viral pathogens is also listed to produce various kind of superantigen. For example, retroviruses. Now retroviruses are also of various type, and some of the proteins which are coded by retrovirus can be used as a superantigen, because they can also activate a huge bunch of immune response when they are going inside the human body or process through a particular cell type. If we closely look into the structure, how this superantigen behaves, then we will kind of understand the complexity that when an antigen presenting cell is producing a specific antigen on MHC2. Now the detail of that whole biology we'll study in later lectures as well, but for time being, if the antigens are going inside an antigen presenting cell, they are processed through this MHC2 and loaded over this groove of the MHC2. This MHC2 loaded peptide can be identified by a specific T cell. Now these T cell are having T cell receptor, and the two arms of the T cell receptor are alpha and beta chain. So these alpha and beta chain can bind and evoke an immune response. That's the normal biology actually. So essentially the antigen presenting cell will bind to a specific or present an antigen to a specific T cell to activate the immune response. But in case of superantigen doesn't occupy this groove that can interact with the antigen presenting cell and T cell. However, what they do is they can bind exogenously outside to this loop area. That means the superantigen can bind outside to the antigen presenting cell domain of MSC as well as the TCR complex. So T cell receptor and the MSC chain will eventually be bound with superantigen exogenously, and therefore we see a non-controlled proliferation of the immune cell and also multiple cytokines. So this is just an important way to see that antigens are identified, and there are category of antigens which can evoke the immune response, but these antigens are quite different, which can create a cytokine storm. So these are quite dangerous antigen when it comes to human health. Well, we'll move on and see when we say that if a particular antigen is immunogenic, then suppose if there is a chain of, for example, say there are 100 amino acid in a short chain of protein, how we can say that which area is antigenic determinant. So to prove how many amino acid or the length of the epitope is important, we need to identify few of the things, and then we'll see how it can identify a particular antibody. So the experiment which people are doing in order to determine the typical size of an epitope, the optimal size of the epitope, is what we call Cabot experiment. Cabot experiment is actually came from a scientist who discovered it. So it goes in a proper immunological fashion where people can look into the binding affinity of a specific peptide stretched with an antibody, and once it is bound to an antibody, we can identify its binding affinity and say that this is the critical length of the stretch of the amino acid that can bind to the antibody, and therefore we define the size of the epitope in that way. How it has been carried out traditionally, so what we can do is, in the first stage, the antigen could be denatured. So denaturation stands for the proteins are mostly in the complex form. So once we will treat the antigen with some kind of an denaturing agent, the antigen become, or the protein becomes stranded, right, straightened away. So once it is a straightened way, we can see that if we can cleave that out in a smaller fragment by a proteases. So we can cleave that protein by proteases after denaturating. So the first step is to denature the protein and then to cleave the protein into smaller fragment. Once the smaller fragments are out, then we can do a size exclusion chromatography to remove a smaller to the larger fraction of the peptide. That means we can segregate a different fraction of the peptide, say smaller, little bigger, and the larger size, and by fractionating it out by size exclusion. And once it is excluded, so before that, what we could do is we can generate an antibody from the whole protein. So suppose, so this antigen can be inoculated into an animal, and when this animal is inoculated with this antigen, can raise an immune response, and we can isolate the antibody. These antibodies are basically produced in response to this protein. So this protein is likely bound to this antibody. That's well known because these are produced in response to the antibody. Now what we did is we fragmented this protein into smaller fragment and then segregated out into smaller fragment and the larger fraction by size exclusion, either gel filtration or the chromatographic technique. And once we incubate this antibody here with all those columns, we could see that if there is a reactivity, then we can say that this antibody can bind to this antigen. That means we can indirectly check the binding affinity of the antibody with those smaller fragment to the larger fraction of the peptide. So if we draw a graph based on the binding affinity, we can see that the smaller fragments are not at all reactive, while the minimum size which can bind to the antigen is this one, and then finally it gets saturated. That means we can see that this molecular weight of the peptide is optimum to raise an immune response. So this is a very classical way people are trying to see the length of the protein that is very important in order to raise an immune response, or the size of the epitopic region. So this is done classically, and we can look into with a typical example here, hypothetical example. So we can see that there could be a fraction, 15 fraction, say this could be 1 to 15. So we can identify smaller fraction in a tube, and then we can see that the peptide size can vary. So the smaller could be say less than 1 Dalton, and then kilodalton, and then the largest could be more than 30 kilodalton or less than 30 kilodalton. So essentially we can segregate this smaller fraction. So you can see 2 through 3, 3 to 4. So these are all the length of the peptide. And once we are adding the antibody, we are doing an precipitation titer. And once we are doing a precipitation titer, we can see that there is no binding till this phase. That means the size of the 4 amino acid or the 4 kilodalton range is not sufficient to raise an immune response. So this could be discarded. After that, there will be a slight bit of reactivity with around 8 kilodalton protein. So we can see that slight bit of information is coming when the protein size is around 8 kilodalton. But then the high reactivity we can see around, say, 8 to 12 kilodalton protein, while very high reactivity is coming when the size of the protein is exceeding 12 kilodalton. That means this is the optimum length where we can see the typical visibility of the immune regulation, and that could be the size of the epitope. So this is what the experimentally people have done in order to identify the size of the epitopes. Now we will see something else, and since we are trying to discuss some of the interesting facts about the immune system, one of the phenomena which we called oxidative burst or respiratory burst is a phenomena where the macrophage cell or the phagocytic cells, when they are engulfing a bacteria, can destroy bacteria inside. And how it is achieved is what is known as oxidative burst. So we could see that once the phagocytic cell can recognize a bacteria, the bacterial cell can be engulfed in a small phagosome-like structure. So these are the small vesicle where the bacteria can be entrapped inside the cell. And once the bacteria is entrapped, then we can see that there could be an assembly of NADPH oxidase. So this is an enzyme which is responsible for doing this oxidative burst. So once the component of NADPH oxidase come into this phagosome area, they can start doing a phenomena where the NADPH actually causes generation of superoxide. Now superoxide is actually coming from the derivation. It's a derivative form of the oxygen and it's a nascent form of the oxygen, which is also called as reactive oxygen species because they are highly reactive in nature. Now once they are produced, there are many ways it can be generated, and after its generation it can be converted in the second step into two major components. One is hydrogen peroxide, H2O2, and that's basically require an enzyme called superoxide dismutase, SOD. Superoxide dismutase, and superoxide dismutase can convert the free oxygen, the superoxide, into hydrogen peroxide, which in turn is converted into the smaller fraction into hypochlorous acid by myeloperoxidase enzyme. So this is another enzyme that's present in the lumen of the phagolysosome. So basically, once the bacteria is engulfed into the lumen of a phagolysosome, there is a series of cascade which could lead to generation of superoxide, which starts from NADPH oxidative complex, and once it is converting superoxide, the superoxide is in turn converted into hydrogen peroxide and hypochlorous acid with the help of SOD and myeloperoxidase enzyme respectively. And once the HOCl is produced, basically HOCl is very toxic and they can degrade the bacterial inside that small vesicle. Now since this vesicle is an entrapped cavity, it is not allowing anything to escape out, and therefore the cell is actually protected while the whole material inside that vesicle are getting destroyed, and that's how the bacteria gets cleared from the system. And once it is done, it is thrown out from the cell as a garbage bin kind of thing. So essentially it's a very important phenomena where reactive oxygen species generated by the macrophages or the phagocytic cells can kill the microbes in a very specific way so that to protect the cells from the untoward, unwanted effect and all. So this is another very important phenomena that happens in the cell type which can do a phagocytosis, the one who can engulf the pathogen and start processing it out. So this is one such phenomena. Then there is always a concern when people are talking about the different types of cells which are available in the blood. One such cell which we have discussed in the earlier classes is called basophil. Now basophil is actually a name given to the cell type which can be stained by the basic dye. They contain a lot of granules, and once they are activated, they can produce a specific kind of chemical called histamine. And once there is more production of histamine, people start getting allergic reaction. And to control, people are taking anti-allergen such as antihistaminic tablet. So a similar type of cell exists in the body called mast cell. The function is same. They can also produce huge amount of histamines and can cause and participate in the allergic reaction. But there is a difference between the basophil and the mast cell. So what are the differences between these two cell types which we need to understand in a proper way? So first come is the origin. So both are originated from the bone marrow because the precursors are same. So their originating point is bone marrow. The long bone marrow, the shaft of the long bone, is what is the actual site of their production. Their maturation is basically happen in a different place. So the basophils are generally coming into circulation, so their maturations are taking place in bone marrow itself, while the mast cells are actually coming into the outside area. So they are mostly in the free spaces. So the connective tissue area is actually the places where the mast cell can be attaining the maturity. They are generally not available in the circulation. They are present in the specific tissue sites, while the basophils are mostly the part of the flowing blood. So they are generally in the circulation most of the time. They generally, the one which is there in the basophil in the circulation don't have a capacity to proliferate because their numbers are fixed, while the one which is there in the bone marrow can do a proliferation if required, because their numbers may change because of the presence of different cytokine at the site of the connective tissue. In terms of lifespan, mostly the WBC lifespan is generally less than a week or in some days actually. So the life of the basophil is also in few days, while the mast cells are little longer lived, so they can sustain their lifespan into the tissue space so that they can do an effectivity to a larger extent when it comes to its requirement. They can express receptor, both of them can express the receptor which can bind to IgE. So basically they can carry the receptor for Fc receptor. So we know that this is actually the antibody. So this is the short arm, this is the long arm. We also call this the heavy chain, and this is the light chain. So we can see that if there are receptor present in the cell type, they can give an access to this antibody. So this is an Fc receptor that can allow the binding of the antibody to the surface, and these receptors are present in both basophil as well as in the mast cell. In terms of granular content, as I discussed, that both are responsible for different kind of histamine secretion. So both can secrete a large quantity of histamine. In addition to histamine, they can also secrete two very important cytokines such as IL-4 and IL-13. So these are the hallmark of the cellular type, particularly basophil and mast cell, because these two interleukins such as interleukin 4 and 13 are actually the identifying feature that can tell us that these cells are actually participating in allergic reactions. So these are some of the facts which we would like to discuss in future as well, and this we will eventually use when it comes to the application of the immunology in total. Thank you.